Thank you again to the speakers, and hopefully we'll facilitate some good discussions. We've got several questions coming in. I'm going to start off, first of all, with really builds off of a question that came in, which was centered around the notion that have the receptors for SPMs been measured in different ethnic groups to determine if there's variation, particularly for those that consume very little to no fish oil. And I preface this by saying that we know that at least with omega-3 supplementation, there's particularly from, say, Skeet Chilton's group and others, to show that there's genetic variations in fats, for example, is important. My lab's actually looked at SPM receptors and seen genetic variation there. And so I'm curious, is that a consideration, particularly, Julio, when thinking about the clinical trials and thinking about genetic variability and efficacy of SPMs may vary across populations?
That is a good question. I mean, what I would like to say that it's true that there are genetic variation, depending on the genetic consideration with regard to the to expressions of these SPMs or the receptors in the immune cells, which today, up to today, I mean, the only supplementation study, they have been kind of pivotal studies or really short course studies, so we haven't been able to show that. But this is a common thing that I would like to, to stand out or to highlight is that they are perfectly available. They have the same route as compared to other fats containing EPA and DHA to be absorbed and to get into blood, and they are quite stable. So the very good thing as compared maybe with the final SPMs, which are the ones who are converted into the immune or in the immune cells to resolve medicine and protective. We have a very short life. I could say that they are perfectly available independently of your race.
Okay. Anybody else want to comment about genetic variation in SPMs?
No, I think this is actually a very good question. And I think this is also we're moving into more precision medicine and into not just with omega-3s, and this is really now maybe with the SPMs as well. When we look at the receptors, also the enzymes, of course, and we have not much more information, like you mentioned, on fats and the more upstream genes, that is. So I think this actually is a very good thing to move into and for precision medicine and SPMs.
And I want to add, there is also evidence of sex difference on resolution. So the women actually perform more resolution compared to men.
Okay. Good point. Thanks for bringing that up. Next question is, are vegetarian sources of EPA and DHA as effective as animal sources in the production of SPMs and the resulting dampening of inflammation and pain intensity reduction? So somebody wants to take a stab at that and clarify some of that.
Actually, and this is something that, of course, we are interested in. The study they have done with the supplement, which is coming from fish oil, and they are present naturally, these SPMs, the monohydroxylated SPMs in the fish oil. But what we have seen that in algae oil, they are present as well. So it's true that there is not studies in human beings, at least, in which this has been shown, but people like ourselves who are working heavily on that.
Okay, thank you. Are there any side effects to watch out for with these supplements?
I wouldn't say that. In fact, I mean, we have the product comes with a self-affirmed grass, and actually, when you look at when you look at fish oils or algae oils, and you measure properly these monohydroxylated SPMs, you could see that there are plenty of them. So, I mean, we could see that we are not exceeding the amount that people could have in their own diet. Xavier de la Rosa, Dr. de la Rosa, has said before that there have been studies in which they have been given up to 10 grams of these supplements to animals, to rodents, and they haven't seen any side effects. I would like to say that the data, they are supported by all the safety data of the supplements or fish oil or algae oil containing EPA and DHA, but we know that they contain also SPMs, and so we could not see or foresee any side effects in taking these supplements.
Okay, a couple of questions here have come in. Just a quick one here. I assume the answer is no. Is that the new dietary ingredient? Is this considered a new dietary ingredient by the FDA or GRAS? And I assume the answer is no. Julio, that's probably a question for you.
Yeah, well, actually, they have been in the market for a few years. This product comes with a self-affirmed GRAS status based on the process that we are obtaining then, and the natural occurrence of these compounds in fish oil and algae oil, so that there are not any safety or I would say worry with regard to them. On top of that, that means they have been considered by the European Food Safety Authority, or one of the national entities of the European Food Safety Authorities, that they have natural equivalence as compared to fish oils.
Okay, and along those lines, what do you think about algae as sources for EPA and DHA for SPM, as precursors for SPMs, and are there concerns about fish oils and toxins?
Well, that's a debate which is today, and I must say that we are trying. We have a process in which we are trying to take out all the toxins which are coming from fish oil. I mean, we don't use raw fish oil, of course, and we are trying to give a very efficient, but at the end of the day, very lean process to enrich these fish oils or algae oils with this compound. So I would say that, of course, again, fish oil and algae oil, they contain this monohydroxylate SPMs, and the way we are processing that is the same way that we are processing fish oil or algae oil, which are present in the market for I don't know how many years, and so I don't anticipate any problem with regard to this.
Okay, I'm going to shift gears here a little bit, go a little bit more biochemical, and this is a question that popped up that essentially says, what seems to be the main reason why the enzymes that produce SPMs from omega-3s are so inactive or inadequate? And then in people versus animals, I'm not sure I get the in people versus animals part, but there are studies out there, I mean, that come to mind in which it's been shown that it was not an omega-3 deficiency or lack thereof. We can have discussions about how much omega-3 you should have in your diet. That's a whole nother argument. But that there were potential, for example, with the 17-HDHA story, and I believe it was with leukocytes, where it was shown that one of the LOXs was inactive or not turning over as much. So can you comment a little bit about that in mouse studies in humans? Maybe Xavier, if you want to take a stab at that, or Hildur.
Well, I'm not very familiar on that work, actually, so I think I'm going to step aside and give it to Hildur if she can.
I know, I mean, I haven't really gone deep into that but we know, of course, it's not just DHA and EPA that are substrates. So in that study, was it completely inactive, the enzyme, or was it just different substrate fluxing through? Because that can also be a problem.
Yeah, it wasn't complete inactivity. I think it was, just reduced activity, and the question is why. Exactly.
And what we know about the enzymes, that depending on the cellular localization, they can have different activities. So, and that can depend upon stimulation of cells. So if you have a stimulation of the cells, you change the sublocalization, for example, 15-LOX or 5-LO, and that can change how it's going into the pro-resolving mediators or the pro-inflammatory, especially with arachidonic acid that can actually go in both directions.
Okay. And then there's a follow-up to this question is I was thinking about this with mice. How do they convert ALA better to longer chains, but also SPMs perhaps? So I think the question is centered around, if you supplement with SDA, ALA, does that give rise to good SPM levels in mice versus humans?
Okay, so you want to go from the C18s. I'm trying to find the question.
So we know that I guess the question is, if you took ALA, would you get high SPM levels in a mouse or a human?
I don't think we have seen that. I don't know. Maybe you, Ross, have been working on that. I know you've been working with ALA.
I have not seen anything with ALA. I know rates of ALA conversion to EPA DHA are vary, but are generally low, and so.
No, exactly. And I think nobody has studied today taking ALA and then looking at the SPMs, like the downstream, if you want to look at then further converting into DHA and then the SPMs. But then we can also talk about what about the octadecanoids, which are the oxylipins from ALA and LA from the C18. Or these prosomal mediators, we don't know. Like, there's no bioactivity known for these mediators. So we don't know what they do actually.
Looks like somebody's commented there was a paper with ALA and mice supplementation. They found higher SPMs in the colon in the context of colitis. So definitely something to follow up on.
Absolutely. I'll definitely check that out.
Interesting indeed. In fact, if you want to put the reference in there, people can look it up. I'm going to shift gears here to another question that's come up. And this is one that I feel like at every omega-3 or every SPM meeting, and this is a contentious area, is omega-3 versus omega-6. And the question is, omega-3s have been reported since 1971 in Lancet. FDA has approved 4 drugs to treat heart disease. The ratio of omega-3 to omega-6 is very high. And have you checked the ratio of omega-3 to omega-6? And maybe this question is directed towards you, Hildur, and some of the work that you were presenting. Has that been looked at? Could that be a driving force?
It's actually a very good question, and definitely something we are looking into. And I'm sorry, I can't really comment on that at the moment. We just have to wait for the paper when it finally comes out.
I think Chris Ramsden's group had published a paper in PLEFA recently that had shown in a rat model that high omega-6 was potentially lowering metabolites coming from omega-3. So I think there's some suggestion of that in the literature, at least preclinical literature, right?
Yeah. Absolutely. No, there's absolutely indication of that. But particularly in our study, I can't comment on that.
Also, is there any comments about what would be the possibility that omega-3s or omega-6s, and again, Hildur, this goes back to you, I think it was, there was a paper that showed that derivatives of linoleic acid, which, linoleic acid is very abundant in the Western diet, most consume PUFA. Is there a possibility that maybe it's not the actual fatty acids, but it's metabolites from linoleic acid that could potentially impair, say, infectious responses?
Absolutely. And there are some indications for that. We're working on some data on that as well.
And in your study that you have going on or is in the process with COVID, is there any thinking about timing of supplementation and particularly with some of the stuff that Julio was presenting with timing?
I mean, of course, timing is everything, like when to start. But like in our study, we take patients that are coming in to the hospital. So they usually have had COVID for some time and they're presenting severe enough that they're coming in. I mean, for us, we don't really then we start treating. They come in, they're admitted, and within the 24 hours, if they consent, they start the treatment. So we like to get them as soon as they come into the hospital. So hopefully in the beginning, they're getting, so these are not ICU patients, so these are not the most sick patient, but bad enough. And in our study, we ended up getting more geriatric population by default. I mean, we were going to include everyone over 18, but ended up having those that were included I think the average age was around 80. So we have this geriatric people, so these older people coming in with they need oxygen, they need to come into the hospital and we start treatment.
I have another topic to bring up that I think is an important one. And I encourage the attendees to please enter more questions. But what I'd like to bring up is storage pools, and certainly that there's in the literature there's good evidence of eicosanoids and being in the esterified or unesterified pool. Any suggestion with SPMs? Are they sitting is there a possibility that they may be sitting esterified and stored that way?
Yeah, we've definitely seen at least the monohydroxy acids. That's for sure.
And how stable are they?
Yeah, maybe Julio, want to take that?
No, you would like to go?
It's fine. I mean, I don't think I haven't seen the study that's actually looked at the stability, well, from what I've seen, especially from what I saw in my breast milk work, is that they seem to be very stable in the breast milk. So somehow they are being protected. So for me, that suggests, and we didn't really go into that, and I think this is something to need to be followed up with, like how and why are they being protected there? So they are in some kind of a pool there within the breast milk, within the lipid nodules.
Yeah, and the per se esterification would actually protect, and yeah.
And definitely, I mean that the fact, for instance, the monohydroxylated SPMs, and they in fish oil, in algae oil, or in the supplements that there is in the market, I mean, they have a very extensive shelf life. So they are very, very stable indeed. They are esterified. They are actually in the same form as compared to EPA and DHA, mainly in TGs, and they are digested, absorbed, and going into the blood the same way as compared to fatty acids. So somehow, as Hildur is saying, that they are protected, and the best way to be protected is probably in the esterified form. They are treated, or the body treat them as the same way as compared to fatty acids once they have digested and absorbed.
Okay, thank you. Another question that came up earlier that has come up in different ways has been dosage. And I think it's probably best for us to think about dosage for prevention and dosage for treatment. And that's obviously will vary quite a bit because, and it'll vary based on sex and age and many other variables. But generally, can we think about SPM, intermediate dosing? And Julio, maybe if you want to take that question about what is appropriate dosing.
Yeah, well, these, and I would also get Hildur and Xavier talking about that. The vast majority of the study has been done with with resolving medicines, protecting that we know that they have very short half-life, as Dr. De La Rosa has said, and they are needing a very, very low amounts to exert their actions. So it's true that today we do not have a lot of literature with regard to supplementation and long-term supplementation studies, or short-term in terms of acute inflammation. What we have been seeing is that they are available. They go to their tissues, and in animal models, it has been shown that they are effective and low doses, because we had to remember that there are very few amounts of SPMs which are converted in resolving medicine and protectins, which are active at the levels of nanograms or picograms in the cells or in the affected tissues. So that's one thing. Of course, there are at the moment, I can tell you, and Hildur was talking about some studies we had to show efficacy at different doses. But we have shown, and we are doing a clinical trial that there are doses, we are talking about 2 grams or something like that, of this supplement, which seems to be effective.
Okay. Great, thank you.
So if we have to give a number, like how much as a dose, the paper from Patricia Souza that she published on Research Circulation that Julio showed, they were using 4 grams as a therapeutic agent because they were reprogramming the host immune response. So I would say maybe, and it's a big if, maybe for therapy, 4 grams would work since they proved that, and then maybe 1 gram to 3, that would be kind of a dose for prevention, more or less.
Okay. So another topic's been brought up, a really interesting one, and the questions are: there omega-6 derived SPMs, so they're referring to the lipoxins, and how does SPM derived from arachidonic acid counter the effects of SPMs from DHA, EPA? And then I'll ask the second part after that. So maybe you guys can talk a little bit about the lipoxins and how omega-6 derived versus omega-3 in that relationship.
Yeah. Okay. So it has to be known how they are synthesized. So it depends on, as Hildur was mentioning before, the lipoxygenases. It depends where they are localized in the neutrophil, for instance, if they are in the cytosol or they are anchored in the nucleus, and then they will trigger the synthesis of SPMs, or by the other opposite end, 5-lipoxygenase can do LTB4 as well, which is very pro-inflammatory. So you have to look where is the actual enzyme that it's synthesizing these SPMs, not only the end product.
I'm familiar with the nuclear localization story with LTB4, but does that also hold true for the lipoxins as well?
No, I haven't tested. I don't know.
Okay.
So for the 5-LO actually to take part in the lipoxin formation, you want it to be in the cytosol.
Yeah.
So that's usually where it meets the 15-LO and makes the lipoxins. And then upon stimulation, the 5-LO locates to the nucleus, and that's where it meets FLAP, makes 5-LO. That's kind of usually we see. And then you can actually have different genetic variations of 5-LO that it is more located in the cytosol. And those people tend to have more lipoxins than, for example, LTB4.
Okay. And the second part of that question is, I asked this question because infant formulas can contain DHA and arachidonic acid and could have opposing effects, especially in infants with atopic conditions. So if anybody wants to comment about opposing effects of DHA and arachidonic acid in the context of infant formula. So I think the question is really directing at a broader question that often arises is, arachidonic acid is often in the literature you'll see as being pro-inflammatory, and which is not entirely correct. And so if you can comment maybe about that broader issue, that there's a balance, right?
Exactly. And like you said, it's often deemed to be the mean guy. It's a pro-inflammatory, it's bad, which is not. I mean, and we need the eicosanoids when we need an inflammatory response, and we don't want to be immunosuppressive. We need this. It's just all about this balance. And arachidonic acid is not bad, but we also need to have DHA, because DHA and also EPA, they give rise to more families of SPMs, while arachidonic acid has more of the pro-inflammatory end, but it also has the lipoxins, the pro-resolving mediators, which are in the family with the other DHA and EPA SPMs, so has the same or similar characteristics effects. But of course, each of them have individual actions then on top of that. But they all have these main actions of limiting eicosanoid or pro-inflammatory mediators and stimulating phagocytosis and nephrocytosis. These are kind of all of these SPMs have these key mediators. Yeah, so sorry, go ahead. No, I was just saying, so having both DHA and EPA, it's all about the balance. And like you said, and I think also it's about the condition. So when you introduce it into the system, if the system is healthy, it's not going to be a problem. And what we saw, for example, in breast milk, you have prostaglandins, you have thromboxane, you have leukotrienes, but you have much higher balance of pro-resolving mediators. It skews very much towards, so you have very high reservoirs of SPMs in the breast milk. You have all the others, but the balance is skewed very much in the pro-resolving end. But this changes in breast milk from mastitis mothers. So if you have an inflammation in your breast and you have mastitis, then it's opposite. Then you have very high level of pro-inflammatory mediators indicating of the inflammation.
Thank you. I think the question often arises is there excess consumption of omega-6s, and can that be a potential driving force toward chronic inflammation? I think part of the answer there is it's left unchecked due to low SPMs, but this is something that always arises. If anybody wants to comment on N6-PUFA consumption in the diet and its role with inflammation.
Well, historically, it's been changing the ratio of intake in the diet from omega-6 and 3 from 1 to 1 more or less in the history,
right.
And now more or less like 1 to 16. So of course, maybe it's not that we are taking excess of the other. It's like we are taking a deficit in the omega-3s. So by amending this ratio would be more helpful. In terms of resolution.
And as Dr. De La Rosa is saying, that the fact that you are having a different intake based on the diets that we have nowadays, or changes in the diet nowadays, is that both omega-3s and omega-6, they share the same enzymatic pathways to be converted from alpha-linolenic acid to EPA and DHA, and linoleic acid to arachidonic acid. So if you have more substrate on the omega-6 areas, you end up having more arachidonic and less EPA and DHA subsequently. And then that's the reason by which it has been shown that maybe supplementation would be good.
Okay. I think we've got just a couple of minutes left here. There's a question in here about, would you recommend taking SPM to someone with Crohn's, and if yes, should it be more than 4 grams a day?
Well, again, all these studies need to be performed first, and then, because this is measuring 4 grams orally intake and then measuring the levels of SPMs in blood. And they see this reprogram of the immune host. But in Crohn's disease, that specific, I don't know if actually it would be helpful only 4 grams or it would be enough or not. It needs to be performed, this study, yes.
Mm-hmm.
Yeah, there's, again, that means, as it has been shown, that there are people who have been doing IBD models using DSS or TNBS in order to look at the efficacy, especially when we are talking about pure resolvins or protectin and medicines, but there haven't been any studies with a supplementation, a natural supplement containing monohydroxylate, SPMs, even in animals, and of course not in humans. One could say that, of course, the inflammation and autoimmune diseases, we can talk or hypothesize that they follow the same pattern in the resolution of the inflammation, and then it is common sense to think that they will be part of the solution. But again, that means there's no studies unless in human beings to show efficacious doses.
Okay. Final question. We have 2 minutes left. And again, thank you all. A pleasure to hear all of you speak and comment. Any idea if the trace minerals and vitamins that exist in oily fish help SPM production from EPA and DHA? And conversely, if the lack of these nutrients may hinder SPM production?
Well, that's a good question, but that's a whole career.
Yeah.
No, it's a really good question.
Yeah.
But I don't think we have looked into that yet.
Yeah.
But for sure, if these trace minerals are affecting the enzymes or somehow helping them skewing one way or the other, that would be definitely a whole PhD and whole career, like you said, for someone to look into.
I'm going to start thinking about nutrient interactions.
Yeah, exactly.
Yeah.
And absorption and all, a lot of.
Any last minute comments with about a minute left that anybody'd like to make?
No, I want to thank everybody. Thank you, Hildur and Julio, and the audience, and for you, of course, Dr. Shaikh. And thank you, the audience, as well.
Yeah, well, thank you for all the questions that came in from the audience. Hopefully we were able to answer all of them. I think we did. And we just have a few thank yous in here. But with that said, look forward to the next one, and we will see you sometime in the future, hopefully in person and not virtually.
Yeah, definitely. Thank you very much indeed. It has been a great pleasure, a great honor to be here. Thank you to the audience, to our chairman, and of course a luxury to have participated in a symposium where Dr. Arnardottir and Dr. De La Rosa are speaking.
I'd also like to thank you all. I mean, it's a great pleasure of everyone for listening, and to Ras and Julio and Xavier and everyone in ASN.
Great.
Thank you.
Thank you very much.
Thank you.
Bye-bye.
Bye.
Bye.